I'd like to welcome Prof. Siebert. Thank you very much for being here tonight. We are really excited to hear about this topic. So Prof. Siebert has been part of the SAAB community for quite a long time, and he's he's made valuable contributions. So he's always had an interest in geobotany. He started his early career at the South African National Biodiversity Institute, where he managed a capacity building program. And then he moved on to the academia and has been teaching botany for the past 20 years. And he's currently a professor in botany at the Northwest University, where he's got a great lab of postgrad students that also supports the SAAB conferences regularly. And they focus on the plant and soil ecological interactions, especially in harsh environments. And so they've had numerous research and travel grants, including ones from National Geographical Sciences and Horizon 2020. So they've been really making large footprints in this world globally. And we're really looking forward to hear what their work has been about. So yeah, Prof. Siebert has been part of the SAAB community since 2001 and has also contributed to organizing one of the SAAB conferences, has served 2 terms on SAAB Council, and has been also on the editorial board of the South African Journal of Botany for 13 years. So thank you for your contributions to this community. It's much appreciated. And now we would like to hear more about the work that you want to share with us. So it looks like the screen sharing, everything is all set up and working. So please go ahead, Prof. Siebert. Thank you very much.
Thanks, Anina, for the nice introduction. And good evening to everyone listening in tonight. I must say, I feel quite fortunate. It's not every botanist that can say that they give a SAAB webinar once in a super blue moon. So that's pretty fantastic. Also to the SAAB community, I would just like to say thank you once again for the medal. I think I feel very honored to have received it, and really appreciate that you have recognized my contributions to the field of botany. And then also thanks for the opportunity to present something tonight, to talk about my research. In fact, I was wondering what I should be talking about, because the SAAB Silver Medal is more for your contributions to the development of botany in Southern Africa, not really so much, I guess, for excellence in research. But yeah, I thought it would be nice to talk about some of the research I've been doing in geoecology, because it's a neglected field, and it's something I'm quite passionate about. And I guess it also shows a bit of the journey that I've been walking since I became a botanist, and how that's also influenced students and young people and staff members, and how obviously that has then built botany further in South Africa. I'm not going to be too detailed in the scientific side of it, but I guess I want to tell the story. And because also geoecology has, for the largest part of my career, been more of a sideline than a main focus. People probably know my work more in terms of socio-ecological systems and agroecosystems, and how that influences biodiversity patterns within grassy biomes. But on the side, I've been doing a lot of geoecological work, and more so nowadays. And I guess that's what I'll be doing for the next 15 to 20 years until I retire. So I'm very, very excited about the opportunities that are coming my way at the moment. Would you like me to keep the video switched on, or should we just use the screen? Anina, is that...
Yes, you can keep it on. That's fine.
That's fine. Right. Okay. Right. As all botanists probably know, botany goes way back in your life, and that's me in the 1980s. in front of my own little garden. When I went on my first play dates, I always found it quite interesting that other kids didn't have their own plants. They had dogs and cats, and I soon realized that this wasn't a common way of common for young kids that age to already be gardeners. So I soon had to get myself a dog to fit in better. And then over the years, obviously, I've chased this dream of being a botanist, and that's the other picture shows me in 2022 in the Namib Desert. After many, many years of being a botanist, and how fortunate we are as botanists to do what we do, getting out, seeing a lot of places, working with plants, a very exciting field to be in, right? So, and it's a very joyous experience, in fact, to have that as a career. So just to quote Vickers du Plessis after all his heroics this past weekend, he always says, "Hulle weet nie wat ons weet nie." They don't know what we know. And I think that's also the same for botanists. Other people don't really know what we know, and why we always look so happy and so excited about things is because they don't know plants, right? Okay, so since my childhood, and maybe, I mean, more so at university, one of the things that I always wondered was, why do plants grow where they do, right? So people take things as it is. It's, in fact, most people just say that's where the plant is. And as scientists, we always want to know a little bit more. Now, with me working in socio-ecological systems, a lot of where plants grow in urban environments and in agricultural environments, people actually determine where plants grow. And obviously climate and soil, etc., plays a role, but people can change those environments to suit the plants, right? So there are other drivers within anthropogenic systems. But when we look at natural systems, you all know it's a very complex history and a whole lot of numerous environmental factors that determine whether a plant can grow where it actually grows. And one of the things that's very intriguing to me is all the different kinds of soils and how that gives rise to different plant communities. Now, we all know about climatic limits. If you just need to travel through South Africa to know that you can have anything from desert through to grassland to savannas to forests. So the fynbos down in the Western Cape, and a lot of that is due to climatic variation that we have at a regional scale throughout the country. Back in 1916, John Bewes published a paper in Journal of Ecology, and he said to use soil as a basis of classification for South African vegetation would hardly succeed. And he was referring to British ecologists using soil to define the plant communities on the British Isles. Now, obviously, the British Isles have a very homogeneous climate and didn't leave them with much else, I suppose, tiny islands, and they used soil and topography to classify their vegetation. So Bewes was right in a way, because you can have different soil types, different geologies with exactly the same vegetation, but a lot of this is obviously scale dependent and very much linked to our biomes in terms of the climate. In the 60s, there was a scientist, Hiram Wild from Zimbabwe, who did some, and Zambia, who did some amazing work in studying the effect of soils on vegetation. So one of the interesting things that he found was when he worked in areas with similar climate and there were serpentine soils present, suddenly the woodlands and savannas would disappear and everything would change into grassland. So as soon as he walked onto the granite and directly adjacent to the serpentines, he would be walking into woodlands. And that intrigued him, and he wondered why. And one of the reasons that he put forward, obviously, was that the soil having very high levels of metals and very high levels of magnesium within those soils. So he was one of the first people that said, right, there is definitely a nice substrate limitation that we can see within a climatic region. So I always say, I wish I could have known Hiram Wild, because I'm probably the person in the world that's read his papers the most. So yeah, it's a lot that I learned from the early work that he had done. Now, this is a picture of an area. This is what we refer to as harsh soils. I will get to harsh soils a bit later. At first glance, this is in Sekekuniland in Limpopo province. You would probably think this is just erosion due to overgrazing and overharvesting of trees. But the very interesting thing about these areas are that when you look back at some of the older works written by the missionaries in the Maandagshoek area, in Sekekuniland, they're already in those, now we are talking 200 years ago, 250 years ago, they already refer to these areas within their work, stating that there are these vast areas of white exposed soils without any vegetation. And so these areas have been around for quite some time. These are not recent changes. And there's a very good reason why these areas look like it's due to the type of soil that's present that is being generated from the geology in this area. And these soils have very low levels of nutrients. They've got high magnesium levels, got high levels of metals, especially chromium. So yeah, these soils are what we would call harsh soils. And this is also the area where I did my PhD. So it's pretty well known in South Africa already that soil and geology can contribute to habitat diversity, and because of that, to species discontinuity. Now, Schmidl has done some excellent work, and the research group have done some excellent work in the Western Cape and Northern Cape, where they've looked at quartz fields. And they've done some really nice experiments to show how the changes in soil lead to changes in species composition. And I think that's proven a lot of, or supported a lot of the theories surrounding the soil-plant interactions in Southern Africa. But even back in the 18th, Skoll, in a book on ecology of tropical savannas, has already shown that within areas with the same climate, that the only reasons that can be put forward for changes in savanna vegetation is usually due to the soil and the geology that is present in that specific area. And anyone that goes to the Kruger National Park will know, as you travel from east to west, the vegetation changes as the geology within the national park changes. It's a very nice area to go and look at changes in vegetation due to soil variation. So what is a harsh soil? So internationally, they often refer to the harsh soil syndrome. It's that keeps certain plants or most plants out. So these soils don't have a lot of nutrients. They either have very high or very low pH levels, lots of potentially toxic elements present in the soil, which are available for plant uptake. The soil is usually quite shallow and doesn't have a good structure or texture, which is not very good for plant growth. And then obviously all of this leads to bare open habitats because there are very few plants, and in turn, again, that leads to high evapotranspiration. So if you think back to that slide I showed of the soil in Sekekuniland, you could note that there wasn't a lot of plants present in these harsh environments. So how did the plants respond to this? So there's one group of plants that will be, well, the majority of plants will be completely intolerant. They will just not be able to grow on these soils. So they are excluded completely. Then there's a second group of plants. We say they are plants that are broadly tolerant, so they can grow on or off of these and germinate and develop into adult plants. And then we have species that only grow on these soils and they don't occur in the matrix outside. So they are completely restricted to these harsh soils and they are endemic to them. In this specific photograph that I have on the slide, there are about 5 to 6 Sekekuniland endemic species that are growing down there in that little valley, and which even includes small trees and shrubs. So some plants are completely what we call edaphic specialists. They've adapted to survive in these environments. Now, harsh soils are very common in Southern Africa, obviously, and across the world. Here are a few examples. We have kimberlites on the top left. Top right, we have gypsum soils. The bottom, we have gypsum soils on the west coast of South Africa and Namibia. Metalliferous soils that you find all along, especially all along the Bushveld igneous complex. And then on the bottom right, you have serpentinites of the Barberton greenstone belt. And these are all, these soils all meet the criteria that I've mentioned on the previous slides that define harsh soils. And one thing that they share is stunted vegetation, sparse vegetation to survive in, and the ones that do are highly adapted to those types of environments. Now, there's been a couple of pioneers in Southern Africa looking at these harsh environments, people whose work I've studied in detail over the years and who informed a lot of the work that I did on my PhD. I've already mentioned Hiram Wild for the work he's done. And then, of course, Kevin Balkwell has been doing, for many years, been doing exceptional work on the serpentinites. Bram van Wyk has done exceptional work all over Southern Africa, looking at areas with anomalous geologies and the associated species. And then Jolanta Meshas-Prizobilovic from Stellenbosch University, who's done a lot of work looking at plants growing in these harsh environments and if the types of metals that they take up from toxic elements they take up and where they store them within the tissue. So this is an example of some of the work. On the top left is Jolanta's work looking at metal uptake and storage within plant tissue. Kevin Balkwell's done a lot of work on floristics and describing new endemic species. Wild has obviously done a lot of work on floristics and then looking at vegetation patterns to explain or looking at the soil to explain the vegetation patterns that he sees along the Great Dyke and other areas in Zimbabwe. And then, of course, the work done by Bram van Wyk, which looked at floristic endemism and showing how a lot of the endemics in Southern Africa are actually associated with areas that could be considered harsh, having harsh soils and geologies. So my PhD research, which I did back in the day, it feels so 99. Back in 99, I was fortunate enough that Kevin Balkwell sponsored my attendance of the third international conference on serpentine ecology that was hosted in Kruger National Park. And there I met my fellow roommate, and we became very good friends over the years, Prof. Nishanta Rajakaruna, who's also a geoecologist. And it's very important, I think, I guess a nice lesson for students that attend conferences is that is to mingle and to make friends and to learn. Because back in the day, when we were younger and very inexperienced, it was our connection was based on mostly on friendship. And with the years, as we've done our research and grown in our respective fields, obviously we started working together more and more and more, which has opened lots of doors for both of us. So in 2012, he contacted me because at that point I was doing a lot of research in other fields, not really in geoecology. And he said to me, "Don't you want to do some geoecological research?" And I said, "Wow, yeah, sounds like fun." And he arranged that I could visit Susan Harrison, who was the leading serpentine ecologist in the world at that point. And I spent some time with her in Davis in California. And another lesson for students, if they don't know about it, the KIC fund of the NRF is very valuable, especially if you want to bring people to South Africa. So we applied, and not long after that, we were able to bring Susan and Nishi to South Africa. And they just published their latest book, Serpentine, the Evolution and Ecology of a Model System. So it was very nice to be able to get these two authors to South Africa, spend a lot of time together doing research, and then sitting down and thinking, what is it that's needed to put geoecology on the map in Southern Africa? So we came up with these 5 things. We said, right, we need to expand geoecological work. More work needs to be done. We need to start thinking of how can we use harsh soils as a model system to test ecological theory. We should also just not look at plants. We should not forget about lichens and biocrusts, because this is was an open field at that stage, and anyone working on that would be do some of the first research in that specific field. There was a lot of talk about trophic level transfer of metals at that point, and we thought that would be something nice to do, a first study for Southern Africa. And then we had to show what will we do with all this information. So what are the restoration applications of this type of research? Then in 2015, so obviously we did a lot of all of that work, and in 2015, my wife and I got bursaries from the Northwest University to go and do a sabbatical in the Netherlands at Wageningen University, and both of us had to obviously develop research plans and papers, etc. And I decided to focus on geoecology because, I mean, my other research fields were running nicely, and because of my very strong interest in geoecology, I thought it would be very nice to develop a focus for myself. So I came up with this aim. I said, right, when I go back to South Africa, I want to understand how chemically and physically harsh soil shaped the diversity, ecology, and evolution of plants, lichens, and biocrusts. Right. So very excited. I went back and started to put things in place to do more and more research in this specific field. While I was at Wageningen, I attended a conference in Malaysia a couple of years earlier, and that was based on research that I had started doing with some of my colleagues and also with Nishi. And we published 2 papers during the time there. And what was interesting about the 2, we were just exploring, but with the first paper, the geobotanical investigation, we said, right, we can see patterns of vegetation in the Vredefort Dome. But we wanted to know, are these patterns associated with the underlying geology and soils? So some curiosity science, and we roped in a geologist and we looked at it, and of course, yes, very nicely, every time the vegetation composition changed, the vegetation composition and structure changed, we were on a different geology. So the Kudusfontein complex is actually a very nice area to work because of the short distances that the geology changes. Sometimes just over 20 meters, you can move onto a new geology. And then we said, right, but what if we can't see any differences? How does it affect things that we cannot see? So then we worked on mafic and ultramafic substrates in South Africa, and every time we could see that the geology changed, we would sample the soil crust and then go back and look at whether the algae in the soil changes. And yes, of course, very nicely, some very nice results. As the geology changes, so does the algae in the biological soil crust. Obviously, it's not specifically related to the geology itself, but due to the habitat that's created by the specific geology and how it breaks down into specific soils in terms of the texture, etc., etc. So with the one we could see the differences, the other one we couldn't, but every time it came down to the underlying soil, leading to a change in diversity of and structure of what you see on the ground. So while we were at Wageningen, Nishi came up with a nice plan. He said, let's go and apply to National Geographic, and we can get a nice project running, do an expedition to collect lichens on and off of harsh soils. And this expedition would then involve 3 old guys. It was myself and Nishi and Alan Friday, and then 3 young up and coming students from universities in the US and South Africa. And we would then go on an expedition, collect these lichens, and in the Barberton area, and then also in Griqualand West. And then we would analyze the data and see if there are any differences in the composition due to changes in geology. Right, so from that, we came up with a whole lot of papers. Here are some examples of the things we did. Interestingly, going through it, basically the same topics as I did in my PhD. So not really anything new, except that we worked in different areas and we looked at different life forms, but finding in most cases the same patterns, and but basically doing the same thing. So in a way, we did address our research agenda, but we were not able to really move up a step as we wanted to start looking at the harsh soils as model systems. So the progress with the research agenda at that point wasn't too good, but at least we could take 2 of them. We've expanded our geoecological work, and we've now included lichens and microbes in our research. So we were very excited about that. So we found some new species of lichens. We found we did more detailed studies of biological crust, comparing also other microbes within the soil. We found a new hyperaccumulator of nickel, and we defined plant endemism in Griqualand West as part of the larger study. So although nothing was new in terms of the research approach, a lot of these papers actually gave rise to some great things. So our community ecology work on biocrist was actually the first that was done for serpentines in the world. So it actually put us on the forefront of being able to study biocrusts on serpentines. The bioaccumulation paper caught the eye of people globally and led to the development of our PRXRF group that are looking for hyperaccumulators, which I will talk about a bit later. The taxonomical work looking at the lichens on serpentines brought a whole lot of top lichenologists from around the world to South Africa. And to this day, they still come. They don't necessarily work on serpentines, but they work in other parts of South Africa. And as you all know, lichens tend to most of the time grow in harsh environments. So it's very nice to have brought lichenologists to study the flora in Southern Africa. And then the studies on floristics has opened the doors, catching the eye of Scion, and they've recently appointed a PDP, a postdoc, to further study these floristic patterns in harsh environments, looking specifically at banded ironstone and dolomites. So in 2017, I started to establish a lab because I was noting that I was being able to do more and more work in this field, and we were getting out papers, and I thought it would be very nice to have it more student focused, so training students, building the capacity of geoecology in Southern Africa. To make it stronger, we linked up to the California Polytechnic State University's geoecology lab, and that improved obviously the quality of the research that we are bringing out, sharing of supervision duties between universities, and also teaching exchange at the universities, and that strengthened the students in terms of their exposure to research, and then also improving the quality of the supervision and research that we are doing on this side. We've received a lot of bright support over the years to keep this going, which we are very thankful of. And just before the talk, I thought I wanted to just check numbers. to see how well geoecology is doing in terms of my own research. And I found that now 2 of the 4 postdocs that I've had have been geoecologists. 15 of my 34 master students have specialized in geoecology, and 3 of the PhD students. So whereas 15 years ago, 70% of my research was in other fields. Nowadays, it seems like it's improving. So I'm very excited about that to focus more and more on geoecology. Then in 2018, it was quite nice to get an invitation from the European Union. Asked me to come and attend their first gypsum ecosystem research conference in Turkey to give an overview of gypsum research in Africa. So obviously, gypsum is also a harsh soil, which had me quite excited because it felt like they are starting to notice the work that we are doing in terms of harsh soil environments. And there I was elected as the African representative and asked to organize a Southern African expedition, and then to also coordinate secondments of staff and students from Southern Africa to travel to Spain to get further training in this field. So in August 2022, we hosted the expedition in Namibia and South Africa, and we had 28 participants from 7 countries attending. And we did a lot of workshops in South Africa and also in Namibia, and came up with an agenda for gypsum ecosystem research in Southern Africa. And so basically our first effort to use harsh soils as a model system for future research. And this agenda was published in the South African Journal of Science a couple of 1 or 2 years ago. And we also did a lot of surveys, where ecological surveys, which are modeled and repeated all over the world. So all of the data that we are generating goes into an international database that will be asking a whole lot of ecological questions on the ability of plants and biocrusts to survive in these environments. Obviously, nothing from that's been published yet, so all I can say at this moment is watch this space. Hopefully in future I'll be able to give a talk maybe at SAAB about the work that we did during this fantastic expedition. So the research agenda was developing nicely. We could now add number 2 and say, right, we've now established harsh soil as a model system. So we were progressing well, but we were not really getting to the trophic level transfer and restoration applications of our work. So COVID kind of brought a whole lot of things to a standstill. As you all remember, we were basically all sitting at home and hopefully writing papers and reports and getting all of those things off the desk. But I've been very thankful since then for a couple of young scientists that's shown interest in geoecology and has done some fantastic work with me and the rest of the lab. People who've really made a great contribution to where we are hoping to move. So they've done a lot of applied research. One of the things that we've been looking at is metal pollution in the environment, especially in areas with harsh soils that are rich in heavy metals. And we wanted to determine, right, what are the conditions under which these soils pollute the environment, right? So obviously we chose to work near mines where there's a lot of activity, and we looked at and we found that different land uses and different topographies have different levels of impact in terms of the creation of dust in the environment, which then end up in soils, people's gardens, and then also in plant leaves. And we also found that the contamination with certain elements does happen, and these pose a health risk to people in those specific environments. So we looked at the levels of metals from the surrounding environment that end up on plants that are used, that were considered useful plants that are used for medicines and food. And we found very high levels that do have a health risk if they are not washed properly before they are used. Then we also started writing up a long-term project where we were looking at the restoration of kimberlite tailings where we've had some major successes. On the picture on the right, you can see kimberlite tainings that has been rehabilitated with probably about 15 to 20 species growing there, all native from the surrounding environment. We did a lot of experiments over many years looking at which species can be germinated in these soils, which of the native species are in fact able to grow on kimberlites, so naturally grow on kimberlites, and also looking at the combinations of using topsoil with kimberlites, et cetera, to restore these environments. Because you must remember that at the level, the height above sea level in these areas is about 3,400 meters. So it's really alpine, and the wind blows the whole day. They have snow even in November. So it's very harsh environment. And then on top of it, a very harsh soil environment. So it took many years to find the species and the recipe that enabled us to get plants to colonize these slopes. And I guess you can go and read these papers if you want more information on that, because it kind of explains the whole process in detail. Then we also started looking at phyto extraction, how to use plants to remove toxic metals from the soil. We've been doing a lot of work in finding these plants, because only 6 hyperaccumulators are known from South Africa, for instance, in our whole flora of 21,000 species. So we are looking to use portable X-ray fluorescence to go to a area and to try and find species by scanning them to see what are the metal content within these species, and whether we will be able to use them, in fact, to be able as phyto extractors on mine dumps. And then we've also gone and done some greenhouse tests where we've used plants that have shown potential, looking at their ability and under what conditions they are able to accumulate, what conditions they are not. So if a plant is a hyperaccumulator or an accumulator of metals, it doesn't mean it always does accumulate, because the conditions need to be perfect for it to happen. We recently had an international PXRF workshop, inviting experts from across the world to come and attend and to develop a research agenda, which we will hopefully share with people quite soon, on using PXRF to discover plants for phytoremediation, not just in Iberia, but also going out into the field and scanning wet material. The progress is doing quite well in terms of our where we wanted to go, except the last one that was still outstanding is the trophic level transfer of metals, which is a very interesting field and something that we've always wanted to look at. Okay, so what have we done in terms of the theoretical part? So in a global study that involved a lot of the countries in the world that have serpentines, we looked at plants that are able to survive under stressful conditions. And we asked the question, when they are on these harsh soils, and we used ultramafic substrates as an example, do these plants have a different specific leaf area? Because we knew that if it does, it shows that these plants are slow growing and they are probably drought resistant plants, because those are character traits that are associated with such plants. And we did the study, and yes, very significantly, we found that serpentine plants actually have the ability, or sorry, not the ability, they have the characteristic of having narrow, thick leaves, which gives them a very low surface leaf, specific leaf area. We're also looking at the drought tolerance hypothesis, where we are suggesting or hypothesizing that plants that are adapted to, physiologically adapted to survive in dry areas will also do well on serpentine. So one of the first studies that we did, we went on a secondment, colleagues and I went secondment to Spain, where we did a lot of the analysis and write-up of the work. data that we collected. And we found that, yes, although fungal diversity was lowest on dry serpentine areas, on the wet serpentine areas, there was the diversity was much higher. And also in areas that were not serpentine areas. So this showed to us that there's no drought tolerance effect for microbes in the soil in terms of their ability to survive on serpentine. And a lot of the results showed that the soil was actually just too toxic for a lot of the plants. And together with drought, was one of the drought together with toxicity was the reason for the fungal diversity decline. So, but we are sure, or kind of speculating, that this won't be the same for higher plants, and it's a part of future research that we are doing, where we are looking at whether drought-tolerant vascular plants are better adapted to survive on serpentine. And then finally, we managed to get a good student looking at the trophic transfer of metals from the soil to the plants, to the herbivore, to the predator. And so far, the student has submitted a paper which has been accepted with major revision that will hopefully appear in future, where he just did a review. And in the review, it showed quite clearly that metals are transferred from the soil, plant, herbivore, predator, and which then obviously means these metals do move through the food chain. But what we found was, because of the different harsh soils and conditions that people work in, either in the greenhouse or out in the field, the methods are quite diverse and varied in their approach. So we need to standardize the methods to be able to compare results that are currently found when people are studying trophic transfer. And the student is finishing his studies this year, Tian, and he will be publishing the results that he found from his own study in terms of the transfer of metals through the trophic system. Yeah, so looking at the research agenda, again, we're quite happy. We finally reached number 4, and it's a work in progress, and hopefully there will be some great results from this specific study. So what is the way forward that we are hoping to achieve with the geoecology? We are now part of a global project where we'll be doing greenhouse trials to test various plant soil climate hypotheses regarding plants and harsh soils, which is in collaboration with the United States and also Spain. So hopefully in the future, we'll be able to give some nice results for that. We're also doing further greenhouse trials to optimize the remediation of contaminated lands, where we test different species and known hyperaccumulators and non-hyperaccumulators, and seeing if we can use them either for phytoaccumulation, that is taking up metals from the soil, or just phytostabilization. So those are plants that don't necessarily take up metals, but they can tolerate them in the soil and thereby bind the soil in these degraded environments. And then we've got a big project scanning specimens from Southern Africa and Iberia. To date, we've scanned nearly 1,000 specimens already. And we are looking to find more hyperaccumulators of metals, and then to develop a database that we will make available for mining companies, etc., to use if they want to know which plants they can use to clean up any polluted areas. And then we want to further conduct plant biocrust and soil surveys across South Africa, or Southern Africa, in fact, and to look at underexplored areas that have harsh soils, and hopefully find some interesting species and also new plant communities in these areas, to understand the whole geoecology of Southern Africa a little bit better. Right, so one cannot do research without people that enjoy the same things as you do, and a lot of people that I'm very thankful for in my life that have done a lot of research with me over these past couple of years, looking at geoecology, and also creating lots of opportunities for me to live the dream and do what I enjoy most. So to these people, I just want to say thank you and thanks a lot, and looking forward to great collaborations in the future with students. So yeah. And then also to my wife, Frances, who most of you know. Yes, she's always part of everything I do. Like, I'm always part of all the research she does and all the ideas that she shares. So it's really lovely to have someone in your life that loves plants even more than you do. So thanks, Frances, also for all the work and all the good times that we have botanizing. And then thanks to the audience, I thank you for the time that you've given me to share some of my research. And then also to all these institutions that make it possible, because nothing can happen if there's no financial support for things and also equipment. So thanks, special thanks to all these institutions. Thank you.
Thank you very much. That was fantastic. We appreciate that very much. The floor is now open for questions. So please feel free to put a question in the chat box or just raise your hand. I am having a bit of technical issues, so Adiemi or Elizabeth, if you see hands that I'm missing, please just let me know. But I'm going to try to keep an eye on everything. There we go. There's one question by Tumbetsu, who's asking, what are the main functional trait differences between intolerant, indifferent, and endemic species?
Sorry, can you hear me, Anina?
I hear you, yes.
Okay, I was just worried I was on mute still. Yeah, thanks for the question. So, yeah, there's a lot of trait differences that exist between species. So, for instance, Siebert, I know about your own research. So, one thing that you will find interesting is that plants that are tolerant of these very harsh environments, except for the fact that they've got low specific leaf areas, that they invest a lot of their resources in the underground structures. So, especially in the roots. So there's a grass that we use for restoration on kimberlites, and it's called Tenaxia disticha, which is a high altitude grass. And after 3 years of growing, this grass would have about 3 or 4 leaves above ground, not longer than probably 5 centimeters. And we were worried that it wasn't growing properly, and we then dug up these grasses to see maybe we could find the reason for it. And when we dug them up, the biomass underground was about I think it was about 24 times higher than the biomass above ground, with some of the roots extending 50 centimeters into the soil. So these plants they do a lot of investment in their roots, for instance. I don't know if that answers part of your question.
Thank you very much. I'll go on to the next question from William Bond. On the trophic impacts, do mammal herbivores avoid plants on harsh soils, such as ultramafics? If not, could one use the trace of the distinct elements to trace large mammal impacts on distributing nutrients?
Yeah, so that's an interesting question, because a lot of work has been done, especially by Bob Boyd from the US, where he's looked at how plants actually use some plants actually accumulate metals in their tissue to deter herbivores, because they're not sure whether it's a taste thing or whether the animals can actually pick up on the metals, the high metal levels in the tissue. I assume it would be a taste, and so they would eat a little bit and then move on. Especially insect herbivores prefer to avoid plants that have high metal levels. There's even they call it the elemental defense hypothesis. Kevin Balkwell has also been doing research on that in the past.
Thank you. I'm going to take one more question in the chat, and then there's a hand up. So there's a question from Dr. Heidi Hawkins asking, she's curious about the hyperaccumulators. If they are slow growing, do they require a lot of time to remediate heavy metal source and mine tailing?
Yeah, so that's very important. So that's part of the study. For instance, South Africa has got a superstar plant called Berkheya cordii, which grows on these harsh soils. And what makes it so fantastic is the fact that it is a herbaceous plant that grows extremely fast. And because of this plant growing at such speeds, it's very valuable in the rehabilitation of mine dumps when they want to extract metals from the soil. And so one of the things that we are looking for is we are looking for plants that are able to produce a lot of biomass very quickly. And obviously, that's quite a difficult thing to find. And that's why when plants like Berkheya cordii come along, obviously they are considered very, very valuable by the mining industry, for instance. So yes, the idea is to find the ones that do grow faster, the ones that are well adapted to the conditions and have certain traits that enable them to be highly productive plants.
Thank you. Then Helga Knutse, please go ahead and ask your questions. You can just unmute yourself, please.
Thank you. Hi, Stefan. Can you guys hear me?
Hi, Olga. Yes.
Yes. Thank you. It was a really interesting talk. I was just wondering, as you know, I did a lot of all of my studies in the sodic zones, and
Yes.
sodic zones also have a lot of characteristics that you mentioned that these harsh soils have. But do you consider them to be harsh soils, or if not, why not?
Yeah. So one could, but I think, but you can correct me if I'm wrong. The sodic zone has very high levels of nutrients, right?
Yes, there are nutrient hotspots.
Yeah. So that means that a lot of plants find them quite will find the conditions quite good. So it's not excluding a lot of plants really, except maybe the sodium that's in the soil, high levels of sodium that could be excluding some species. But I guess one could call it a harsh soil, but it's also very nutrient rich, which is the only thing that concerns me in terms of classification as harsh soils.
Okay, thank you.
Okay.
Thank you very much. I think there's one more question in the chat here. Or a comment from Nishi. What a great talk. Thanks for taking us through your geoecological journey. Keep up the great work for putting South African geoecology on the global map and for training so many young geoecologists along the way. So that's just a bit of congratulations for you.
Thanks, Nishi.
Kevin, I see your hand up. Please go ahead.
Hi, I just wanted to go back to what William was asking. So we did some work where we had a senecio where some populations accumulate and some don't accumulate, that's Senecio coronatus. So the same species, both populations on serpentine. So we thought that was quite useful to try and test whether or not the hyperaccumulation stopped herbivory. And we used snails to do that. And this was work largely done by Bob Boyd with assistance from myself and colleagues. And what it was found was that the snails that were on the hyperaccumulating senecio grew much slower and died much faster. And they also appeared to eat less. On the other hand, Mark Stelmans has recorded in Songamvelo Game Reserve that the grasses on the serpentine patches are eaten preferentially, especially in winter. And so his hypothesis is that the serpentine, to some extent, is sweet felt in a sour felt environment, where the growth of the plants is limited to some extent by the metals in the soil, and thus there is more nitrogen available to the plants that do manage to survive there. And so, yes, it seems there are mammalian herbivores that do eat grasses that grow on serpentine.
Thanks, Kevin. Yes, there was a very interesting comment made at the last Serpentine conference, where someone actually noted that, or asked, why is there no research on herbivory of hyperaccumulator plants being done in the world anymore? So I guess that's a field that needs a lot of attention.
Thank you very much for that. Here's one question in the chat from Elaine Slurton asking, have you noticed whether hyperaccumulators tend to be forbs or grasses, or is it a 50-50 situation?
Well, hyperaccumulators are mostly forbs, can also be trees and shrubs, but mostly forbs.
Thank you. All right. Let me just see if I see any other questions that I might have missed. I was curious about it's great to see how the work that you're doing in geoecology, the fundamental sciences, also translate to useful applications to our environmental issues and restoration, etc. And I was wondering where the links are specifically in the agricultural field, especially with a focus on restoring our soils in agricultural systems. If you could maybe just give a few comments about that.
Yeah, so it's interesting in Europe. Phyto mining is becoming a bit of a agricultural activity. They now use a lot of hyperaccumulators in areas that are contaminated by metals, and then growing these crops as cash crops on these contaminated areas for a couple of years to clean them up, selling the dried plant material to mining companies that then extract the metals from the plant material. It's not something that's really quite popular or well known in Southern Africa. It's not something that I know I've seen some talks, especially in the Western Bushveld complex on the platinum mines, where they've been doing things like this to clean up the mine dumps and then also to extract. But in Europe, a lot of the cultivation is on old fields that's high in metals, and using that to clean them up. Yeah. So there's a lot of it's something that could be looked into, with crop rotation, maybe to have for each region in South Africa having a specific hyperaccumulator that you can cultivate for a season, just to remove all the toxic elements from the soil, and then to go back to your normal farming. But for instance, maize is very good at taking up metals from the soil. So they actually do quite well as well.
Oh, that's very interesting. Thank you very much. So one more question here from Tumbe. So again, any work or plan to look at long-term consequences of changing climate on soil properties, plant growth in these environments?
Yeah, so that's where SAAB comes in. We are developing some long-term monitoring studies now with SAAB, looking at these areas with harsh soils and to see what effect climate change does have on them. There's a nice theory out there that we can discuss, but it's all about. So if you look at more tropical areas in the world, the serpentine or the harsh soil endemics are usually restricted to the harsh soil, and you won't find them in the adjacent areas because the competition is just too much. But when you look at the more arid, semi-arid to arid parts of the world, where you have harsh soils, often the plants that are associated with the harsh soils are also able to grow in the surrounding areas because of the lack of competition that they experience in those areas. So I think the species, the endemics of harsh soil environments will obviously be a very good indication of, it will be easy to see how climate change affects them or not. But for that, you would need to have long-term monitoring plots to monitor this for quite some time.
Great. Thank you very much. I think that is the end of the questions. And